Can a heart on a chip replace the real thing when it comes to researching medications? Stay tuned for this actual scientific research study that created a heart on a chip. Hey, and welcome to Science is You. This is a growing channel dedicated to making real biotechnology research accessible and fun for everyone. To support this mission, please subscribe and spread the word. Thanks for tuning in. An important consideration when developing new medications is whether or not they could have side effects. Many compounds can have toxic effects on the heart. So it's important to have ways to quickly and effectively screen drugs for cardiac toxicity before introducing them into people in, say, clinical trials. Testing in animals not only carries some ethical concerns, but is costly, and animals like mice or pigs are very different, of course, from people. So there's a growing need for more ways to tell if a candidate medication might be toxic to the cells of the heart. Instead of doing animal studies, we could look at just what happens to the cardiac cells themselves if exposed to a compound. But cells growing in a petri dish is a very different situation from cells in your heart. In the heart, cells experience a variety of stimuli like stretching, squeezing, and fluid flows. Microfluidics is the study of how to create devices that transport small amounts of fluid. Think micro, small, and fluidics, fluid. In this scientific study, the researchers developed a microfluidic heart on a chip to better mimic the environment of heart cells in the body. The scientists designed a microfluidic device to culture cells on an elastic surface. The device consisted of an aluminum well-shaped chamber attached to a heated base for keeping the cells at body temperature, a see-through cover for taking microscope pictures, and electrodes to stimulate the cells. The researchers didn't just dump the cells into the device, though. They carefully positioned cardiac myocytes, the cells responsible for heart contraction, onto chips. To make the chips, the scientists took a thin cover slip and covered it with a piece of painter's tape. They then cut 4 rectangular shapes into the tape with a laser engraver and peeled off the cut tape using a tweezer. In a series of coating steps, the researchers created a thin film over the rectangles that was peeled back to form tiny cantilevers. To position lines of the cardiomyocytes onto the cantilevers, the scientists use a technique called microcontact printing, where tiny stamps are used to press exact shapes or lines of a substance that cells can stick to, in this case fibronectin, onto a surface. Then cells are added onto the surface or seeded, and any cells that did not attach are washed away. After doing all of this, the cardiac myocytes were aligned as tiny muscle-like tissues down the length of the bendable cantilevers. Then they put this whole chip into their microfluidic device to form their heart on a chip. Having these cardiac myocytes as thin muscle-like tissues on a bendable surface film allowed the researchers to study how they contract, a type of test termed muscular thin film assay, or to put an acronym on it, the MTF assay. Upon electrically stimulating the cells on the chips, the researchers observed that each thin muscular film could contract freely, and they could measure the contraction cycles. They were also able to microscopically observe cellular nuclei, where the DNA is kept, and actin, which is part of the cell's muscle-like machinery. This showed an organized, aligned structure of the cardiac myocytes into muscle-like fibers. The researchers next wanted to test out their heart on a chip with actual pharmacologically relevant chemicals. Isoproterenol is a drug used to treat bradycardia, or slow heartbeat. They used their microfluidic device to flush in increasing doses of isoproterenol and measured the contractility, like a mini heartbeat, of their heart muscle cells on the chip. They indeed measured a considerable difference in contractility, consistent with measurements in rat and other laboratory models that had previously been reported. Now, this wasn't the first ever report of a heart on a chip, but what the authors did here was redesign a chip containing the cardiac muscle cells in the muscular thin film to fit within a fully enclosed microfluidic device. This permits rapid handling and changing out of fluids, and is intended to more closely mimic the environment the cells would experience in the body. So the authors felt this was a considerable advancement. And enclosing the chip in a microfluidic device also opens the door for connecting chips together, to study how cells behave when connected with other organs, sort of like a human on a chip. This system also minimizes having to handle the cells and manually change out fluids, which can cause temperature fluctuations and make the environment less realistic to what the cells in your body likely experience. The microfluidic heart-on-a-chip device would make it possible to continuously perfuse fluids and different drug concentrations across the cells, and more accurately mimic exposure scenarios in the body. Further, the microfluidic device itself was reusable, which is always a win. With every scientific study, there are limitations and next steps. Here, for example, the researchers bring up that the design of the muscular thin-film chips isn't easy to integrate with very complex microfluidic devices, so they focused on simplifying their device to just one compartment. Also, testing this device on increasing numbers of potential medications and getting further validation that the system really can predict response of an actual human heart is an important next step as well. Also, medications generally are not just flushed directly onto your heart muscle, but pass through your gut and bloodstream first. So figuring out how to link these organ chips together might give an even better idea of how a compound affects cardiac muscle cells. What do you think? Are heart-on-a-chip devices the way of the future? Would such devices ever be able to completely replace animal testing? Share in the comments. If you've enjoyed this video, please subscribe and share with your friends to help out our mission of making real biotechnology research accessible and fun for everyone. And remember,